before I start, I would like to say thank you everyone in this room to be patient until very last session. So don't worry, I'm the last person of this afternoon. So please be patient a bit more, 15 more minutes. And I would like to ask everyone to stay here until finish so that we can make a group photo to make our memories. Okay, thank you very much. So let me start with the thanks to our funding sources. So fundamental funding program from the Culicidae University, Thai Ministry of Higher Education, Science, and Research and Innovation, and also Faculty of Agriculture, Culicidae University, supporting me to travel to Pattaya from Bangkok, and Culicidae University Research and Development Institute, CODI, and Research and Lifelong Learning Center for Urban and Environmental Entomology, in short, Real Ento, and also my mentors, Professor Tira Pap, Charul Pap, and also late Dr. Michael Bang, who was my co-advisor for my PhD study. So we haven't discussed for last 3 days about the mosquito-borne disease, and I'm focusing on the dengue fever, which is the neglected mosquito-borne disease. So as you can see in the table, most of the fundings are focusing on the malaria. As you can see at the end of the table, they have the 10 times more higher number of the deaths compared to the dengue. That is why it is quite neglected. However, it is quite serious now with the climate change. So a couple of years ago in Pakistan, with the heavy rainfall, there was the dengue vector outbreak, which is Aedes aegypti, primary vector of the dengue. So if you go to the Pakistan with the recycling center without roof, all the artificial breeding sites are there after heavy rain. So if you pick any of the 2 bottles, you can see the mosquito larvae one of the bottles. So this is related with the outbreak of dengue fever, of course. So every week, more than thousands of patients occupy the hospital bed. So it also quite social expenses and problems. Not only in the tropical area, but also non-tropical countries like France, Paris. Recently, they also have their dominant cases of the dengue fever transmitted by the Aedes albopictus. Person from who never visited other countries, means non-tropical area also having this kind of issue. And South Korea, of course, we are worrying about invasion, but thankfully still have no domestic cases yet. In Thailand, people already know how to avoid the mosquito bitings when they are going to the forest, so they already were adapted for their outdoor activities using the, you can see in the picture, I took it from the villagers from Putoi. So when they go out, they use their mosquito coil and bring it together with their vehicle. And also we have the long-lasting special repellent product in the market. So we can easily see the product in the market in Thailand, and also that one recently got the approval from the WHO, the series. It is very cost-effective and powerful tool to protect the people and reducing the person, the host and vector contact. For the practical reason, I have to keep thinking about the cost-effective way and sustainability to keep the persons from the mosquito bitings. So I found that in Thailand and tropical area has the serious problem with the weed. This water hyacinths fully cover the lake so that aquatic organisms cannot breathe in, or some fishermen cannot use the boat because of this issue. So they send the troops to remove this water hyacinths. And the local community is already very smart. They adapted the recycling this waste plant, transforming to making some goods. If you go to the Jatujang market, the weekend market, you can see many types of the shoes and bags from this water hyacinths. So we may consider the materials to impregnate the special repellent on these materials. So I wanted to look at more the behavioral responses against the special repellent like methoprolutrin and transfluthrin, and also persistency with the multiple washings. I want to see the long-lasting effect of the transfluthrin and methoprolutrin. And finally, I continued my study not only for the toxicity assay but also special repellency assay using the HITSS assay. I will show you the picture with the detailed explanations. So this is the approach to testing the toxicity and also behavioral responses. So high-throughput screening system, in short HITSS assay, developed by the Professor Glico from the Notre Dame University early 2000, and we have a lot of publications using this apparatus. So WHO recommended this apparatus using for the repellency test and also attractant development. So I will show you one by one. First of all, this apparatus using the active ingredient impregnated filter papers, and this WHO SOPs recommended treated the filter paper and dry out 24 hours. However, for the special repellent, which is very highly volatile pyrethroid, we have to be careful with the concentration. If you keep drying for the 24 hours, you can see the percentage is degraded. So I recommend you to use this apparatus with 1 hour air drying time for the filter paper. So this is what I found from the profit analysis with 5 different concentrations, and we can calculate the discriminant concentration for each chemical. So as you can see, methoprolutrin has stronger toxicity compared to the transfluthrin, around 5 times stronger, so that we have less amount for the discriminant concentration with the same mortality. And the beauty of the HITSS assay is it has a wide cylinder size, so the diameter is more than 10 centimeters. So it is enough space to keep the harnesses fabric, which is quite thick. So I could test the different types of fabrics using this apparatus. So for the persistency, I followed the washing protocol. So I wash it using the soap and following the WHO guidelines. I got very interesting result. After 25 times washes, the natural fabrics still have quite same, not significantly different mortality rate for the tropical TFT and NFF. So now we know that this apparatus can figuring out their chemical properties. We found out the methoprolutrin is 5 times stronger than transfluthrin, but we also need to see how this kind of the sub-lethal dose of the chemicals affect the mosquitoes' behavior, because I tested the susceptible and resistance Aedes aegypti, and you can see here. Of course, the susceptible reaches the 100% mortality. However, the resistance from the field population is less than 80%, means already developed the transfluthrin resistance. So we have to consider what if they just expose the chemical but still alive? What would happen? So we should go with the further study on the behavior resistance. So this is the another 2 functions using the same apparatus, the contact irritancy and non-contact irritancy. So basically this apparatus has the same size cylinder, and we can assemble only a single chamber to the 3 linked chamber, so that the mosquitoes have a chance to contact the treated surfaces or have no chance to contact to the treated surface indirectly, because in the special repellency assay, we release the mosquitoes in the middle, and we have the treated chamber and untreated chamber in both sides, so that mosquitoes can make a decision where to fly. So depending on the concentration, the response is also different. So we call it the concentration-dependent responses. So this is the result. As you can see here, with the 5 times higher concentration of the TFT, showing the higher contact knockdown and mortality. That is why the mosquitoes in the treated chamber has no chance to escape, because they already knocked down. So the percent escape is significantly lower than methoprolutrin. However, if we use the special repellency in the middle, the mosquitoes has no chance to contact directly on the treated surface, so that we have the less percentage of knockdown and mortality. Of course, the mosquitoes can escape to untreated chamber. So the transfluthrin actually push to the other side from the treated chamber. So it has very good repellency effect. So this is the special activity index. We calculate the number of the mosquitoes in the treated chamber and untreated chamber. So if the number is close to the 1, meaning very good repellency. However, the methotrexate with the 5 less amount of the discriminating concentration, showing the higher knockdown and mortality. It is because the mosquitoes fly to the treated chamber. Means the methotrexate may have the less the special activity, unlikely the transfluthrin. So we can imagine the transfluthrin treated chamber fully occupied with the molecules of the chemical, so that the concentration gradation sent off the mosquitoes to the untreated chamber. However, the methotrexate, which has higher contact toxicity, may have the less molecules inside, of course with the less amount of the discriminating concentration, so that mosquitoes fly into the treated chamber. So it resulted the higher knockdown and mortality, so that the special activity also lower than transfluthrin. So we can see the patterns from this HITSS assay, quite depending on their concentration. So the mortality, of course, decreasing as the concentration decreases with the distance as well. But we also found out the transfluthrin and methotrexate, the chemical properties, so that the special repellency for the TREP-T is higher than methotrexate. So the conclusion. So the HITSS assay successfully generated discriminating concentrations for each special repellency agent, methotrexate and transfluthrin, and behavioral responses is obviously depending on their dose dependent, and also interestingly, the natural fabrics persisted longer than the synthetic fabric, which is available in the market. However, we have to think more about the mosquitoes survive from the sub-lethal dose exposed. So this is the interesting result. Special repellent exposed mosquitoes increase their number of their eggs produced. It means we don't know why, but they may have some flash effect after the exposure to the chemical. So the female produce more number of eggs. And this another study also saying that after exposure, the female attracted to the ovitrap much more for their oviposition. So we have to think about this concept, the push-pull strategy. Actually, this idea came from the agricultural field, but we have to think whether this is available for the dengue vector control as well. So now we are working on the special repellent and also developing the lures for the ovitraps and host-seeking female mosquitoes. So I conducted some trials in the semi-field greenhouse assay to compare the lure effectiveness. So the BGS trap and also black light trap for the nocturnal mosquitoes tested with the KU lure embedded or not treated. So briefly, I will tell you the result. With the lure, of course, enhance the trap efficacy. So I have only 1 minute, so you may capture this QR code to listen more details on this field study. So this is my plan. So after this study, based on the scientific data, we may support the local community by providing some properties with this kind of the local product with the mosquito management agent. So I will keep working on this study. So in summary, so the push-pull study actually takes some time. So we should keep continuing this study with continuous funding support. So thank you very much, and if you have a question, please. Yes.
So your experiment is look like a tunnel experiment, right? I wonder if you have more allow the mosquito try to feed on host, like put a mouse in one size, because if the mosquitoes are stimulated by the host feeding, so I think they may be more aggressive and try to contact the treated fabric more, because I used to treat it in the bed net and use my arms against the mosquitoes. So without the host, the mosquito just try to escape from the insecticide. But once you have host, and it's really attractive, so maybe the mosquito try to feed more and try to contact more on the net. So it's possible if you apply this insecticide in the bed net, and I wonder whether there will be more mosquito or more motility and more lockdown if you have one host inside the net.
Yes, sir. Thank you very much for your comment, Ajahn. So actually, yes, we also do have the wind tunnel assay. So as Ajahn described, we have the tunnel and release the mosquitoes at the end, and we have the artificial feeding system with warm blood here, the other side. So the mosquitoes will contact the artificial feeding system for feeding. But we also have the control and treatment. So there is the layer of the treated mosquito net in the middle. So we can count the number of mosquitoes feeding the blood. So we can also see the efficacy of the chemical treated net. But this study used the apparatus without any host Q, only for the chemical. So we more likely focusing on the attractant development. So of course, the real human or animal is the best, the gold standard. Even the carbon dioxide, we cannot beat it to developing attractant. Although we still have to keep working on to make it better and better. So not only for the left side study, even though we have very good results from the left side, if we bring it all to the semi-field or real field, the results will be totally different because of the environmental issues. So we have to keep circulating all the process from the lab, semi-field, the field. And from this result, we can learn and enhance more the formula, the mixtures and ratio to increasing the attractant level. And of course, we can add on the real host so that the attractant and repellent interaction can be more specific. So this one takes time and also takes a lot of resources. So we are keep working on it, Ajahn. Thank you very much.
Other questions?
Okay, thank you very much. So now it's time to take the group photo here. So please, everyone, join our group photo. And